In this lecture video, we're going to take a look at what people have done with all of the biotechnology tools that I've talked about in the previous series of lecture videos. And specifically what we're going to focus on is genomes. So after the development of DNA sequencing, allowed us to sequence an organism's entire genome, and that gave birth to a new field of study in biology, the field of genomics. So, here's what we've done with the sequencing technologies. So things really started out with the Human Genome Project, and the Human Genome Project took 13 years to complete. Obviously there are a lot of really great reasons motivating these studies of the human genome. We thought when we started out, okay, if we know our genetic sequence, then we're going to be able to cure all kinds of diseases, figure out all kinds of things about, our own biology. It's going to be really revolutionary. So there was a lot of motivation to tackle this project. The Human Genome Project relied on the Sanger sequencing method, and it used what's called a whole genome shotgun approach to sequence the genome. And I've just illustrated that in this little cartoon here in a nutshell. So basically what that involved is collecting a lot of DNA samples, then chopping those samples up using restriction enzymes. Then those pieces of DNA were then cloned using gene cloning in bacteria to amplify those pieces of DNA, so there's enough DNA to work with to do Sanger sequencing. Then Sanger sequencing took place, and as you can imagine, that was very labor intensive, distributed across a number of facilities. It took quite a long time. And then once you have sequences, what you've got are sequences of DNA that are only 450 base pairs long, but most chromosomes are way, way longer than that. So once you get those sequences back, what you can do is then look for regions between sequences that overlap with each other and use those overlapping segments to then line up sequences and assemble the genome. So again, this was a very labor-intensive international effort. So it took 13 years to sequence the first genome, and it cost, well over $100 million to complete this sequence. So then by the year 2007, so 4 years later, we were down to only 4 months to sequence a genome, and at that point it only cost $1 million. And by the way, at that point, the genome that got sequenced was James Watson's genome, of all genomes. And now, we're using next-gen methods, it's possible to sequence a genome in the space of a single day, and it only costs around $1,000 to sequence a genome. So that's at a point where now it's approaching affordability to sequence a genome, and what that means is that it's possible to sequence the genomes of a much wider range of organisms and get much broader insights into variation in genomes across organisms and also what's shared in common across those genomes. So here's what's happened in terms of the cost of sequencing genomes over time. So this only runs from the 2001 to about 2013, but what you'll notice is it actually isn't really tied to changes in computing costs. So Moore's law is basically predicted drop in cost associated with changes in computing technology over time. And you'll notice this line follows Moore's law pretty closely up until the point where next-gen sequencing really started to take off, and then that dramatically brought down the cost of sequencing genomes. And in addition to being able to now sequence genomes relatively cheaply, we also figured out that you can use these technologies to sequence groups of organisms together. So, the classic thing that we want to do is to be able to sequence a set of microbes. And so when you sequence a group of organisms like that, we then refer to the study of that collection of organisms as metagenomics. And this has been tremendously powerful as well, because we've known for a long time that it's not possible to grow all different types of the microbes that are found in the environment in culture in the laboratory. So this now lets us characterize everything that's present in a given environment and then compare between different environments. And I provided an example of this in my sample Biology in the News presentation, if you've watched that, of comparing the sequences of the gut microbiomes of people eating different types of diets. This is also often used to compare the microbes that are found in different types of soils. So for example, you could compare the microbes that are found in an organic farm soil compared to microbes found in a conventional farming environment. And you'd probably find very different microbial communities in each of those environments. But to kind of get a sense of how genomes get studied, I thought it would be useful to think of a specific example. So I'm going to take an example from some of my own research, which is, let's say that you have, an ant species that you're interested in. It's a new ant species. This one is a seed harvester ant called Pogonomyrmex californicus. So you sequence P. californicus's genome, and now you want to know something about that genome. So for example, maybe you want to know, how does this ant species detect smells? And maybe more generally, you're just interested in understanding, how does the ability to smell different types of things evolve? And I chose smell for a particular reason, in that actually this is an area where there is a lot of really fascinating stuff going on. So this is a set of research questions that a lot of people are very interested in. So this brings us to a really crucial decision that was made over the course of deciding to sequence the human genome. And that decision was that genome data should be made freely available to everyone. So you cannot own a genome. Genomes are available to everyone. And so in making that decision, then people also had to make a decision about, well, how do we make that freely available to everyone? And so I'm going to just give you a very brief overview of some of the tools that are out there that make it possible to access genome data for any organism that's had its genome sequenced. So we'll start out looking at GenBank, which is an online DNA sequence database that lets you search and compare matches for DNA or predicted protein sequences or protein motifs across organisms. And I'll also talk a little bit about BLAST, which is a tool designed to help search through GenBank. And then we'll also take a quick look at a tool called CN3D, which is a tool for generating a three-dimensional view of all determined protein structures. Okay, so here is just a snapshot of some of the major pages within GenBank. It's a pretty technical site, so it doesn't have a very slick-looking interface or anything of that nature. But what it does have is it contains a lot of data. So this little inset graph that I've got on this slide just shows cases where we have genomic data for different organisms, and then also whole genomes sequenced for different organisms. And as you can see, the number of sequences in GenBank has been just growing and growing and growing. It's amazing. So then, if you sequence a genome, it gets deposited in GenBank. And then now you probably want to start knowing something about that genome, because it's one thing to have a sequence of A, T, Gs, and Cs, but another thing to know, like functionally, how does it actually lead to an organism? So that brings us to BLAST, which is one method for starting to answer that question. And basically, what it will do is let you start to find similar regions between different biological sequences. So one of the first insect species to have its genome sequenced was the fruit fly, and fruit flies also smell. So for my seed harvester ant, one thing I could do would be to conduct a BLAST search for olfaction genes in fruit flies compared to my ant species, to see if I uncover any hits, to see if there are similarities in the genes for being able to smell that are present in the fruit fly and in a seed harvester ant. So, I might do that. I might get a hit, and then I might wonder, okay, well, this looks like a region that's going to code for a functional protein product. But what will that protein do, and what might it actually look like? So that would bring us to something like CN3D, which provides a three-dimensional view for a known protein structure. So what I've got here in this little window is basically showing a bunch of amino acid sequences rather than nucleotide sequences. And this is lining up some new protein that I've discovered with motifs that are found in other types of proteins where we have a better sense of what that protein structure looks like. And so we can compare our query against other proteins that are in this database to start to get an idea of what kind of shape it has. So this is just highlighting one particular domain that appears in the protein that I'm interested in. And so that, for example, could help me evaluate whether something that I think is an olfactory gene, whether or not that actually is produces an olfactory protein. Because if it produces a protein that is wildly different in shape from every other known olfactory protein, then that suggests, I might be onto something very different, or I might have a false lead. This also opens up what are called systems biology approaches. For systems biology approaches, biologists they're not just studying and thinking about the function of a single gene at a time. Instead, they're thinking more in terms of clusters of genes. So here's some of the types of questions or problems that get studied using the systems biology approach. So, one question, well, what groups of genes get expressed together and how do those gene products interact with each other? So, with my ant example, that would be saying, well, let's use RT-PCR to figure out all of the genes that are expressed in the ant antenna together and see what we can learn about how that collective gene expression pattern translates into actually being able to smell. As another example, we used to kind of conceptualize of cancer based on the organ or tissue where that cancer is found. So we'll talk about, melanoma as a form of skin cancer, or liver cancer or brain cancer. But as we've become more able to start sequencing the cells that are found in a cancerous tissue, what we've discovered is that, there's no such thing as liver cancer. There's, many, many different types of liver cancer. Each type of liver cancer may very well be the product of a different set of mutations. And cancer generally isn't the product of just a single mutation somewhere in the genome. There are some exceptions to that. Breast cancer is kind of a classic one, but in many other cases, cancer is a product of a complex collection of changes that takes place in cells. And so in working to tackle, problems in understanding, how cancer arises, how to treat cancer, how to prevent cancer, biologists have developed a cancer genome atlas to facilitate the comparison of gene sequences and gene expression in cancer cells versus normal cells. So that's another arena that's really grown tremendously over the past decade. And on top of that, things are now even going a step further from there. So, for a while we were very focused on genomes, sequencing genomes, but, we know that things don't really end at a DNA sequence, right? There's RNA involved. There are proteins that are produced. Those proteins are acting on different substances found inside of cells. So now people have kind of expanded beyond just thinking about a DNA sequence to thinking about these multiple different levels of function within a cell and how they relate to each other. So this is then referred to as multi-omic mapping. We're not just looking at a genome, we're not just looking at a transcriptome, so a collection of RNA transcripts, we're looking across those. and also thinking about some of those epigenetic changes that happen that influence which genes get expressed when and where. So, we've developed tools to also throw in and think about the status of the chromatin, along with thinking about what's happening with individual genes. So again, this is there's a lot of information here to work with. We're in a very information-rich era, and it's incredibly exciting to be doing biology in this time frame, but it can also be a little bit overwhelming as well. So here's just one other example of this systems biology approach. So just last year, what we've managed to do very recently is drill down our methods to be able to sequence the RNA that is present in individual cells. So we can separate individual cells from each other and start to ask, how does gene expression in cell A compare to gene expression in cell B, the cell that's next door? So I found this paper that I think is just utterly fascinating, where in the past we had some idea that different parts of the liver kind of specialize on different metabolic activities, but when these biologists were able to actually separate out individual liver cells and examine the RNA in those individual liver cells, what they found is totally new groups of cells that were very different from each other in what genes were getting expressed. And so, that's really changed our thinking about, how the liver works. It's not a homogeneous, uniform organ. There are a lot of different cells doing a lot of different types of things, interacting with each other within just this one organ, within the tissues of that organ. So, what else can we do with this overwhelming amount of data? We're going to kind of zoom back out a little bit and just explore some of the outcomes from some of this whole genome analysis and whole genome comparisons. So here's 3 sets of characteristics that we'll look at across genomes. First, thinking about just properties of genomes in general. So, how big are genomes in general? What's a big genome? What's a small genome? Along with that, how many genes are typically found in a genome? What's usual? What's unusual? And how dense are genomes? So is a genome just gene, or is there a lot of space and sequences that don't get transcribed or translated in between genes? Those are kind of big questions to get a better grasp of, how we wound up the way we are. Along with that, that has allowed for the analysis of what are called gene families to start addressing questions about how we get new genes. I'll talk a little bit more about that in a moment. And that also opens the door for thinking about the evolution of genomes in general and at large. So when we reached a point where we could sit down and start comparing entire genomes to each other, we encountered a number of surprises. I'm actually going to start with something that's not a surprise, then we'll talk about the surprise. So the table in this slide here includes a list of a number of the organisms whose genomes were sequenced fairly early on, and then we've got information about how big those genomes are, how many genes they contain, and then how many genes per megabase of DNA. So just to get you oriented here, so up top we've got 2 groups of prokaryotic organisms, so bacteria and archaea, and then towards the bottom we've got a collection of eukaryotic organisms, including ourselves right here. And this first column, so this is haploid genome size. This is size of the genome you'd find in a haploid. So for us, that's the genome size in an egg or a sperm cell. If we take a look at these numbers, first if we look at the prokaryotes, these are all numbers between 1 and 5, basically. If we hop down now to the eukaryotes, even yeast, okay, now we're up in a larger numeric range. So yeast has 12 megabase genome. Same thing is true of all the other organisms listed here. So, okay, probably not a huge surprise that prokaryotic genomes are smaller than eukaryotic genomes. But once we get within a domain, so just looking within the eukaryotes, we've determined that there's not really any clear systematic relationship between the size of an organism's genome and that organism's phenotype. And so to give you a really wild example of this, down here at the bottom of this table is a plant, a Japanese canopy plant. So here's the human genome, 3,000 megabases. And here's this canopy plant genome, 149,000 megabases. Enormous. And the photo in this slide is a picture of this plant. So if you just looked at this plant, you looked at its tissues, you really, you would not predict that it would have such a huge genome. But there it is, an enormous genome. Then things get even more interesting when we start looking at the number of genes within each of these genomes. So just think back, this involves knowing something about what a promoter sequence looks like and what defines exons and introns, and just analyzing that to get at least a crude estimate of the total number of genes found within a genome. So when we did that, we hit on another surprise. There's no connection between the total size of an organism's genome and the number of genes that organism has. So, to look at this, again, it's less surprising when you look at prokaryotes versus eukaryotes. We have more genes than the prokaryotes do, right? Prokaryotes are somewhere between 1,500 to 7,500 genes. Here, eukaryotes get up to 40,000 genes. So in this table, the standout is actually rice has 42,000 genes. But then if we kind of look across these organisms and compare the size of the genome with the number of genes, it's really apples and oranges. So something with a relatively small genome might actually have a relatively large number of genes in it. Something with a much larger genome might not have all that many more genes than something with a smaller genome. So it really varies. And along with that, in ourselves, we also discovered that, we thought we had more genes than we actually wound up having. But I'll also note that this particular comparison does not factor in the role of alternative splicing. So this is just total number of genes, and so it's very difficult to incorporate alternative splicing on top of that, because that's just even more labor intensive to actually figure out gene products. So it's going to take us longer to evaluate that across a range of organisms. So it could be that when we sit down and actually look at actual gene products, that we find a very different pattern than what we see if we just look at the total number of genes. So for the moment, I think we can comfortably conclude that we don't see a strong association between genome size and gene number. But beyond that, we can't really make any strong conclusions. And then there is something that happens, though, when we start to evaluate gene density. And that is that there are certain groups that really stand out for having very low gene density. And so the groups that stand out are actually humans and other mammals. So if we look now, the prokaryotes, again, they actually have very high gene density. In the eukaryotes, as we look down this table, humans and pandas really stand out as having low gene density. So what's going on there? So if we evaluate the human genome in a slightly different way, here's what we come up with. So here's a pie chart for the entire human genome. And when we sit down and look at it, only 1.5% of the entire human genome actually consists of exons. Right, so that's 1.5% that is coding for all the proteins, ribosomal RNAs, and tRNAs that are found in a human being. Which, just take a second and think about that. That seems, again, just totally inexplicable when you first start thinking about it. Okay, so then there's a couple of other categories of DNA sequences that we can relate directly to some kind of function. So that's this 25% here that consists of introns and regulatory sequences. So these aren't coding for products, but obviously they're still doing something important in the genome, especially these regulatory sequences, determining which genes are turned on and turned off. But that still leaves us with a whopping 73% of our genome that doesn't seem to have a very clear direct function. So we refer to that other 73% as intergenic DNA, so DNA between genes. And we can classify some of that intergenic DNA to some extent. So here are kind of the categories that we've come up for classifying that DNA. So there's one cluster that we refer to as pseudogenes. That is a sequence that looks like a gene, but when you actually try to chart out what that might wind up producing, you wind up getting premature stop codons and things that just don't really make sense for that to actually be a functional gene producing a functional gene product. There's also a lot of DNA that is very repetitive. So there's actually two categories here for that repetitive DNA. And that includes some repetitive DNA that contains a lot of sequences that we have pinned down to something called transposable elements. And I'm going to come back to that in a moment. And for this intergenic DNA, for a very long time, biologists thought, oh, okay, well, that's not coding for any protein product, so obviously it's not that interesting. So, it's just junk DNA that's just kind of carried along from generation to generation. So that was the initial thinking about everything else, kind of dismissive. As time has gone on, we have started to discover more and more fascinating functions of this non-coding intergenic DNA. So it's not really junk DNA. It's doing something, but we still don't entirely know what it's doing. We also know that a lot of this DNA is very highly conserved. So what that means is that you can find these sequences across a wide range of organisms and over the course of evolutionary time. So there is still a tremendous amount to unpack about how our genome and how other genomes work. But I want to come back again to that point about transposable elements. So what are those transposable elements? So this brings us back to an earlier geneticist, also a Nobel Prize winner, Barbara McClintock, who was really interested in how corn kernels got their color and kind of interested in the genetics behind that. And as she was studying corn kernel coloration, she discovered color patterns that really didn't make sense in the context of how we thought that genes for color should work. Because what it looked like was those genes were randomly getting switched on and off as the kernels developed. And eventually, after scrutinizing these corn kernels for a long time and studying characteristics of the DNA in these corn kernels, she figured out that it was possible to explain what was happening with those color patterns as being disruptions of the color genes by DNA insertions. So what she figured out was that DNA doesn't stay in the same order all the time. There are some DNA segments that can actually move around in the genome. And so we refer to those segments that can move around as transposable elements. And we also call them, for short, jumping genes. A lot easier to say. And at this point, we've figured out that there are basically two main types of these transposable elements. So the first type are referred to as transposons. And these elements have an intermediate stage, a mobile stage that's not in the genome, that consists of double-stranded DNA that basically this will code for proteins that allow this section to get snipped out of this section of DNA, and those proteins also facilitate insertion of this piece of DNA somewhere else in the genome. So, and this can happen either through a cut and paste mechanism, so what I've just described here, or also through a copy and paste mechanism. So this section gets replicated, and then that replicated DNA copy gets stuck in somewhere else. The second major type of transposable elements are called the retrotransposons, and that the word retro might start to make you think about something else. So these are transposons that move around as an RNA intermediate instead of a DNA intermediate. So these rely on the function of reverse transcriptase to synthesize a new piece of DNA and then insert and integrate that new piece of DNA somewhere else in the genome. So this retrotransposon will be inserted somewhere in the genome. It will get transcribed, it will get translated, it'll form, as illustrated here, a ribonucleoprotein complex, so that piece of RNA is kept that gets reverse transcribed and stuck in the genome somewhere else. But basically, this kind of changes our thinking about a genome from something that's fairly static and inherited generation to generation to, oh no, there are pieces that can get cut out and put somewhere else. There are pieces that can get copied and stuck, and those copies stuck somewhere else. All kinds of stuff going on in a genome. So that's a bit about properties of genomes. So now I want to talk for a minute about gene families. So what are gene families? Gene families are groups of identical or very similar genes. So when you look at the sequence of these within a genome, the sequences are extremely similar in multiple locations. And I'm going to talk for a minute about a classic example of a gene family, which is two related families of genes that code for types of proteins called globins. We've actually talked a little bit about globins already. Specifically, we've talked about alpha and beta globins, which are the protein subunits that come together to form a hemoglobin molecule. So the interesting thing is these alpha and beta subunits are actually coded for by genes that are on two different chromosomes. So the alpha globin gene family is found on chromosome number 16, and the beta globin gene family is found on chromosome number 11. And on top of that, there are multiple versions of these alpha and beta globin genes that exist on each of these chromosomes. And depending on how old you are, different members of that gene family wind up getting expressed. So that's also indicated in this picture. This is just highlighting how when an individual is an embryo, one type of alpha globin gene gets expressed and one type of beta globin gene. And then fetuses and adults express different versions of these alpha and beta globin genes, depending on development stage. So, this leads to a question, well, how on earth did this even happen? Our general hypothesis is that there must have been some kind of common ancestral globin gene that then underwent a series of events, so gene duplication, then gene mutation, and then a gene transposition effect to wind up on two different chromosomes. And there's pretty good evidence in support of this hypothesis in that the amino acid sequences found in the alpha and beta globin gene families are very similar to each other. And thinking about that gives us a good working idea, or working model, or hypothesis for thinking about how we could get genes that have new functions. So, how does innovation happen on a genetic level? And this is a hypothesis that's worth continuing to test and explore across as many cases as we can. So, let's compare amino acid similarities between other proteins. And that brings us to a really surprising example of two proteins that seem like they have absolutely nothing to do with each other, but that have very similar amino acid sequence. And those are the proteins lysozyme and alpha-lactalbumin. So lysozyme, so that's an enzyme that's found in lysosomes. And so lysozyme is involved in chopping up bacteria or other harmful substances that try to get into a cell. In contrast, alpha-lactalbumin is a protein that helps to facilitate the production of lactose during milk production in mammals. So it's involved in synthesizing, creating lactose molecules, whereas the lysozyme chop up bacteria. It's very different. So when we compare DNA and amino acid sequences, that gives us ideas about how multiple different mechanisms could be interacting to create new genetic combinations. And the reason why this is so critically important is this is how we think we get the raw material, the original material for natural selection to act on. And we're going to start talking more extensively about natural selection beyond this lecture. So here's some of the major ways that we can get new genetic combinations for natural selection to act on. So one is mutations. We talked about different types of DNA mutations, all the copying errors that can happen. Also through duplication events, also through transposition, and through quote-unquote mistakes that happen during meiosis and mitosis. And there are still a lot of surprises to be uncovered in genomes, beyond this stage. We're learning now that a lot of these repetitive sequences actually do get transcribed in very specific contexts, contrary to our expectations. And we also are finding cases where genes have seemed to kind of appear out of nowhere on a very short time scale that we still can't fully explain with most of our thinking about how DNA gets replicated and how it gets passed on from generation to generation. But being able to make these types of genome comparisons can give us a huge array of insights into all kinds of processes and questions in biology. So, for example, if we compare genomes across distantly related species, that could let us answer questions like, what genes are really fundamental across living organisms? Where can we extrapolate from a yeast cell to a human cell? What do we share in common? Turns out, a surprising amount. It also lets us compare closely related species and ask questions like, well, so humans can speak and chimpanzees can't. And why not? We share a tremendous amount of genetic information. Where are the differences that have contributed to that distinction between us? And then we can also compare genomes within a species and start to study processes like local adaptation. So, this lets us ask and answer questions about which genetic variants are more beneficial within specific environments, and what types of new mutations are the most common in organisms. And just to give you one example of this, I'm going to come back to something I touched on previously, which is, so we've gone out and we're very curious about ourselves naturally, and we've sequenced a lot of human genomes at this point. And in the context of, the genetic diversity and genomic diversity across all the organisms that, we've been able to study to date, what we've determined is that it turns out that humans actually have fairly low overall genomic diversity. So on a genetic level, we're all the same, as I said previously. And putting this in a bit more context, modern humans are only around 200,000 years old. Might seem like a long time, but generation-wise, it's not all that long ago. We've also determined that the highest genomic diversity in humans is found in people in Africa. So, again, the genomic diversity is not evenly distributed across human populations, but we can actually identify multiple human subpopulations in Africa that have high genetic diversity. The biggest sources of that diversity in people come in the form of what are called single nucleotide polymorphisms, also known as SNPs for short. So this is point mutations, just a single nucleotide change between people. And then some other sources of diversity include things like chromosomal inversions, deletions, and duplications. And we've also been surprised by how often we see variation in the number of copies of a given gene. So, there are a lot of copy number variants across human populations. And by doing these studies of human genomic diversity, we've gained some incredible insights into human evolution and questions associated with human health that, again, weren't even imaginable, 20 years ago. So again, it's just really an incredible time to study genomes. And so, there's still a tremendous amount of stuff to be discovered in this arena. At this point, we've already fundamentally changed a lot of how we think about genetics and genomics and evolution. And there's still a lot of surprising new insights to be gained in how and what we understand about how organisms function and how organisms have evolved. So far from biology being, a discipline where we have a set body of knowledge and we know how everything works and nothing really changes, as it turns out, biology is a very dynamic, active field, and there's all kinds of new discoveries on the horizon.